Hi, I'm Jeff Todd, President and CEO of Prevent Blindness. Today, we are, as part of our Focus on Eye Health Expert series, we are interviewing Dr. Mark Pennesi, Professor of Ophthalmology at Oregon Health and Science University, about gene editing for rare eye disease patients. Dr. Pennesi, would you please tell us a little bit about yourself and your area of expertise, and welcome, and thank you for joining us.
Thank you. It's a pleasure to be here today. So I'm an inherited retinal degeneration specialist. So I exclusively see patients with genetic eye diseases, and these are mostly rare eye diseases, most commonly retinitis pigmentosa or Stargardt dystrophy, as well as other genes, some of which can be quite rare, sometimes 1 in a million people. And we are not only interested in seeing patients with these genes to characterize those diseases, but also now we're entering an age of treatments for these diseases. Specifically, we have a number of clinical trials now using gene therapy and stem cells to hopefully slow down the degeneration in these diseases and maybe even improve function.
Great, thank you. And we're here to talk a little bit about a study you've been involved with that utilizes gene editing. What exactly is gene editing?
So I think it's important to understand a little background about gene therapy and how our genes work. And so the analogy I like to make is that our genome is made up of many different genes that code for all the different proteins in our body. And we have about 25,000 different genes. And each one of those genes is like a recipe book that gives you the instructions on how to make a given protein. And you can get mutations in that gene that then mess up the recipe. And sometimes these are very small mutations, such as changing a single letter in the recipe. Other times they can be big changes, like tearing a page out of the recipe book. And prior to gene editing, the approach to fixing these kinds of problems was essentially what we call gene replacement therapy. And that's where we put a whole new recipe book into the cell, in addition to the recipe that has the problem with it. And so in essence, you're putting a new copy in. Gene editing, on the other hand, is going to target specifically that recipe book with the problem, where you go in and you make an edit to your native DNA rather than putting in a new copy. So for example, if you have something, sometimes with a mutation, you can get the creation of a little stop sign that prevents the protein from being fully made. And we can go in and cut that out with gene editing so that the protein gets made. Or we can go in and actually edit a single letter in the recipe to fix the mutation. So that's the difference between gene editing and gene replacement therapy.
We hear a lot about Crispr lately. Is that a form of gene editing, or what exactly is that?
Yeah. So CRISPR really refers to a form of gene editing where what we've done is co-opted a bacterial protein. And it turns out that bacteria in their native state get infected by viruses, just like we can. And they have a very primitive immune system that is designed to cut up the DNA that viruses try to insert into them. And this is actually the CRISPR system. And so they utilize a protein called Cas9, and you can kind of think of it like little scissors that it can cut DNA. And so what we can do is sort of hijack this system for our own purposes and actually reprogram it to make little cuts in the DNA of our DNA. And so that's really just one part of the system. The other part of the system is trying to get those scissors to the place that you want. And so you can think a little bit like DNA, sort of like a ticker tape with code on it. It's a long series of of code. And our DNA has 4 different letters, A, G, T, and C. And so what we can do is almost develop like a little GPS guidance system where we use small pieces of DNA attached to the Cas protein that allows you then to sort of target those scissors to a particular area of DNA and make a very precise cut. And if you have 2 different Cas proteins, you can target off a little region and cut out a little region of DNA. So that might be useful in a situation where sometimes you have a mutation that actually creates a stop sign in the code. So rather than getting the full protein, you only get about half of it because there's a little stop sign in the signal. And so what we can do is design a CRISPR-Cas system that goes in and cuts out the stop sign. And that way you then get the full protein. And that's really the strategy that we've been using most commonly. In the future, there are going to be other ways of editing, as I mentioned, more like a word processor, where we can use the proteins to go in and actually change the letter. You know, for example, change an A to a T or a G to a C. And that's something that we're exploring right now.
Got it. I really appreciate all your analogies. They're definitely clicking with me and helpful. So your recent study has focused on a condition called Leber congenital amaurosis. What is that?
Yeah. Well, Leber congenital amaurosis is a rare inherited retinal dystrophy, but it's a very severe one. So, you know, oftentimes these inherited retinal dystrophies like retinitis pigmentosa don't become symptomatic until patients are in their late teens or early 20s. But Leber congenital amaurosis actually can occur as early as right after birth or, you know, even within the first few years of life. And because it occurs so early and is quite severe, many of these patients will lose vision by the time they're 10 years old. You know, most of them will be legally blind by the time they're 20. Many of them will be completely blind by the time they get into their 30s. So it's a very severe and aggressive form of retinal degeneration. And there are a variety of different types of LCA. For this particular trial, we're looking at one called type 10 LCA, which is in a gene called CEP290.
So this recent study that you're involved in, how exactly are you bringing the CRISPR techniques to bear for treating the LCA?
Right. So, you know, the first component is we have to get the little scissors into the cells. And the way we do that is we actually put it inside of a gutted virus. So we can take a benign virus that would probably cause a mild cold, and we hollow it out, take out all the parts that would allow the virus to replicate or make you sick. And so essentially we're just using the shell as a delivery system. And within that, we put the DNA that codes for the CRISPR Cas system. And then we inject those billions of those little viral particles underneath the retina. And they go and find the target cell, which is the photoreceptor. And they then insert that DNA into the cell, and that DNA goes to into the cell, makes the CRISPR machinery. So it makes little scissors and the guide RNA. Those go to the center of the cell or the nucleus, make the little cut in the DNA, and take out the mutation.
Got it. Got it. So I understand this is the first time that CRISPR technique has really been used directly inside the human body. Is there a reason that the eye made sense for the first attempt at its use, or were you just first in line?
Yeah, so that's correct. You know, so CRISPR has been used in other applications where you can take the cells out of the body, for example, with a blood disorder, and you can take the cells out and put them in a dish and then apply the CRISPR machinery, make the edits, and then put those cells back into the body. That's harder when you want to treat a cell that you can't take out of the body, like a nerve cell. So the eye turns out to actually be a really ideal place to do gene therapy for a variety of different reasons. One is that we have two eyes. So we can treat one eye and compare the results in that eye to the other eye that we don't treat. The second reason is that because we can actually look into the eye and see the retina, it allows us to visualize what is going on. We can very precisely visualize where we're delivering the gene therapy. We can also look for things like inflammation very easily, which is something that we always worry about with these kinds of treatments. And we have a variety of ways of measuring the function of the eyes, such as testing vision or visual fields. And finally, the eye, and in particular, the subretinal space, has a special property in that you tend to get less immune reaction there than you do in other parts of the body. So that reduces the likelihood of inflammation. And I think that makes, the eye one of the ideal places to study gene therapy.
Great. That's interesting. Are there certain individuals who are more likely to be successful candidates for gene therapy?
Well, I think, that's a little bit of a complex question in that, during a trial at any given time, we're looking for different patients. So, very early on in a trial, before we know the safety of a therapy, we're actually looking for patients who are very advanced and don't have much vision, because we need to ensure that the therapy is safe before we go on, say, to children. Now, as the trial progresses, we're looking for patients who might have better vision, because then we're really trying to determine whether or not the therapy is efficacious. And for that, we need people who can do certain tests. And so, at different stages, we have different inclusion and exclusion criteria. We don't, whether someone will respond better or not, that's sometimes a difficult question. And we see variability in every trial. Even with therapies that work, sometimes you see people who respond better than others. And we don't always know every factor for that, but I think it's like that with any kind of medication.
So do you have any sense on the time frame for when this treatment might be more widely available for individuals?
Well, I think that, with any kind of experimental therapy, it takes a long time from when you first treat somebody in a phase 1-2 trial until something like FDA approval. That can be anywhere from 5 to 7 years on average. So, and that's important in that we take a lot of precautions to make sure that the therapy is safe. So even though, we're treating patients now, we go very slow because we want to make sure that we don't see any bad kinds of side effects. And so we start at a very low dose and then escalate to higher doses and then go higher. And then eventually, in a wider population. But because of that, it actually takes, years to get these therapies approved by the regulators.
So I imagine that, the hope is that this sort of therapy can expand to other eye conditions as well. Is that correct?
Absolutely. So, this technology, really, you can think of it like a Swiss Army knife. We've designed it, for this disease specifically, but it's very easy to retool this to address other genetic diseases as well. Not only in the eye, but, in other parts of the body potentially. But, so it's very exciting because it really represents a whole new tool set that we have.
Yeah, Prevent Blindness has been around for 113 years. And And I've been here for 20 years, and just even in that 20 years, we've seen so much advancement, but it seems right now there's just a lot going on for new treatments in eye disease and eye conditions. And it's exciting to be in this field right now.
Yeah, absolutely. I mean, I think we're kind of in a renaissance right now when it comes to therapies for these diseases. And once you get one treatment that works, that just opens the door to so many other possibilities. And it's a little bit like a snowball rolling down a hill. It just kind of keeps getting bigger and going faster. And so we're definitely seeing an acceleration of that. And to the point where now I think we have so many clinical trials, it's hard to keep track of all of them. Compared to 15 years ago, we really had very, very little.
Yeah. Well, as we wrap up, again, thanks for your time. But do you have any final thoughts you'd like to share about the potential of gene editing to improve the lives of those living with these conditions?
Well, I just really think, it's a message of hope right now, because I think that, right now we're targeting one, rare disease, and it probably won't apply to many people, but it's really a big event, because this does open up the possibility of treating many other genes. And we can't treat them all overnight, it takes time, but there's a lot of hope on the horizon.
That's great to hear. And thanks again for joining us.
Yeah, thank you very much.